Hey y'all, so for this session I will be going through the first part of metabolism. It will cover more of the broad aspects of metabolism, and the part 2 session I will be going through the specific metabolic pathways. So I will share my screen with you and we'll be underway. Okay. So the topics I will be going through in this session are the following. Anabolism and catabolism, metabolic diversity, energy and carbon sources, C for carbon, then energy carriers, then transport mechanisms, like facilitated diffusion, simple diffusion, active transport, ABC and MFS transport proteins, and siderophores, and group translocation. Group translocation will play a role in glycolysis. So I will go through these throughout the session, okay? So I guess the first question to really ask are, what are the components of the cell? This is a general question, right? Well, there's small molecules, MLCs. That's just the abbreviation I use for molecules. H2O, inorganic ion, organic substances. Do you know what makes something to be classified as organic? Something is considered organic if it has carbon-to-carbon bonds. Okay. Inorganic is a substance or a compound that does not have carbon-to-carbon bonds. That's like the definition in a nutshell. Macromolecules such as proteins, nucleic acid, carbohydrates, and lipids. Okay. Also, chemicals taken from the environment and used by the cell. And used by the cell. This is the K. So this includes macromolecules, elements, chemical elements, and they're used either for structural components or for energy. Okay, that's the basic overview, okay? Now we're going to be a little bit more specific. Essential nutrients for the cell. We have macronutrients, and basically those are, you think that prefix macro, like large, big, So major, you just kind of, I equate major, the word major with big or large. So major elements that make a cell constituent. Okay. So those examples here would be these elements. I don't know if y'all knew, if any of y'all knew Dr. Chopin, but she spelled her name like this, so you can remember CHOPN, just minus the I pretty much. And then I don't really have an acronym for the potassium, calcium, magnesium, and iron, but if these would be either you can come up with an acronym, or if you just are good at memorizing things in order through rote repetition, that's fine too. Okay, then so we have macronutrients, and then we have micro, the micro prefix meaning small. So micronutrients, and those are usually have the trace elements from the periodic table. They also usually serve as enzyme cofactors. I won't be going into detail on enzyme cofactors. That's something you'll learn more in detail in biochemistry. Okay. So these are the essential nutrients for the cell. And now we're moving on. So now I kind of covered those broad bases with the components of the cell and essential nutrients for the cell. Now with metabolism, you can think of it as falling. You have pathways in metabolism, but you also have branches too, if you want to think of it. Like I think of it as there's two types of metabolism. There's the anabolic and catabolic metabolism. So anabolism, catabolism. So anabolism is more biosynthesis, so you're more like making something, like you're building up something. So build up of nutrients. Subunits needed for macromolecules like sugars, amino acids, nucleotides, it's abbreviated with NTS. Catabolism is more, it's the opposite of anabolism. So if you're breaking down the breakdown of nutrients in order to release energy. And that energy, breakdown of nutrients. To release energy. That energy is produced as ATP. So ATP, I'm sure most of y'all know, is adenosine triphosphate. Okay, I'll be using ATP soon, a little more frequently, especially making sure y'all knew what the abbreviation meant. Okay. Now, I'm going to give you all a little bit of intro to the metabolic diversity of nutritional types. So, nutritional types for different for different organisms. Like this applies to prokaryotes and eukaryotes. The thing is with bacteria, the metabolism or cellular respiration, or you can say metabolism in bacteria or in humans, like both the prokaryotes and eukaryotes, they still undergo cellular respiration or photosynthesis. But what matters is, especially in respiration, if it's anaerobic respiration, which means the respiration is occurring without the presence of oxygen, or aerobic respiration, where respiration that oxygen is required for it to happen. But I'll get to that in a little bit. So this is more of an intro. Organisms need they absolutely need these three elements, which are carbon, hydrogen, oxygen. CHO, if you want to think it that way. Organisms also need electrons. I'm going to denote electrons with that E raised with that negative sign. And then, so electrons provide energy to perform work of the cell, the transfer of electrons provides a way to reduce molecules for biosynthesis. Way to reduce molecules. And those of y'all who have taken Gen Chem, I'm sure the redox tower is coming to mind. Remember oil rig? Oil rig. Oxidation is loss of electrons. Reduction is gaining of electrons. Or you do the Leo Kerr thing, but I learned it as an oil rig. Okay. Organisms thus require sources of carbon, energy, which is usually in the form of ATP, and electrons. Okay. Now, I'm going to go specifically more into the different nutritional types. So there are four. I'll just go about one by one. Metabolic diversity. Okay. So the way you look at this with the different nutritional types, you look at the carbon source and the energy source. So carbon, I'm going to divide this. So carbon source and energy source. I'm just going to do the more broad branches here for carbon source. I'm going to draw another table in a minute, and it'll be more specific on if it's like photoautotroph or chemoorganoheterotroph, that kind of thing. I'm just trying to ease y'all into it, so I'm not throwing these big words at y'all all of a sudden. So the carbon source for an autotroph. An autotroph is basically an organism which can use carbon dioxide as the only or principal source of carbon. Okay? So basically, autotroph, you're thinking the carbon source is going to be CO2. And an autotroph does do carbon fixation. If you're fixing something, if you're fixing a compound, carbon fixation, nitrogen fixation, you're normally saying that you're converting something that's inorganic. So in case with carbon fixation, you're converting an inorganic carbon to an organic carbon. Okay. So like here, CO2 is considered inorganic, and an autotroph, an organism that's an autotroph can convert the CO2, the inorganic carbon, to an organic form. So basically, you're reducing this because, yeah. Then we have heterotroph here, and a heterotroph requires preformed carbon compounds. Okay, preformed carbon compounds. So heterotrophs are organisms which use reduced preformed organic molecules as the carbon source. Okay. Now we're going to talk about the energy sources. Okay, energy source. We have photo and chemo. So photo means energy from sunlight, and chemo means energy from chemicals. Normally you'll see photo or chemo as prefixes for the different nutritional types or modes of nutrition. That's another way to classify this. The chemicals can be organic and inorganic. This will make more sense once I draw y'all my other chart in a minute. So metabolic diversity, this carbon source and energy source, these are needed for biosynthesis, for movement, for biosynthesis, movement, and nutrient transportation. And the sources for this, for the carbon source and energy source, are organic chemicals like glucose, or inorganic chemicals, or like mainly all three of those though. Okay, now I'm going to draw you on my second chart that pretty much includes the energy and carbon sources from different groups of prokaryotes, as well as examples on top of that. I want to clarify something real fast though. Electron sources, this makes sense with the chart as well. If the word litho, that root word's in the nutritional mode type, that means inorganic donors, organo / organic donors. Okay, I just wanted to clarify that before. Oh, and by the way, the suffix troph means nutrient matter, or an organism with given nutritional requirements. Yeah. So there's two side notes here. Okay. Learning some of these Latin root words are prefixes, suffixes, it is helpful sometimes because there are so many of these words and you're like, you just don't want to confuse them. Used by different groups of prokaryotes. Okay. So we have type, energy source, carbon source, and examples. So this first one is photoautotrope. It is the primary producer. The one with the degree sign that I indicate that as primary. The energy source is sunlight because you see the photo prefix, and then carbon source auto, right? You're thinking for autotrophs, the carbon source is CO2. And examples of photoautotrophs are cyanobacteria, specifically Anabaena, Microcystis, and oscillatoria. Next we have photoheterotroph. And that energy source is sunlight as well. But you notice that root hetero, so the carbon source is organic compounds. And the thing with the photoheterotrophy, and I'm not saying the examples I'm giving you, these aren't like all of them, but these are common bacteria that you will see or you'll probably use in the lab or you'll be exposed to at some point during the course. With the photoheterotroph examples, they're the rhodos. Both of them have the prefixes rhodo. Rhodosudo. A-S. Rhodopseudomonas. Yeah. And then Rhodobacter. Okay. Then we have chemolithoautotroph. Yeah, kind of long. Sometimes you might just see it as chemoautotroph without the litho root, but the litho root does help you to recognize, oh, okay, the carbon source is CO2, and the energy source is inorganic chemicals, such as ammonia, nitrate. Okay. And then examples, so we have the rhodos for the photohetero, like hetero, rhodo, and then with chemolitho, you have Thermus aquaticus and Pyrolobus. So Thermus aquaticus and Pyrolobus. Okay, so this thermo, that prefix means heat, and pyro, that prefix usually indicates either, I'd say, yeah, I guess you could say heat, but normally if you see the word pyrogen, that's indicating like you're going to have a fever, so you can just associate these two words with heat, and that you can group that with chemolithoautotroph. So So we're starting to get into the material where in order to keep track of everything, you want to be grouping things either by root word or association, or if you come up with an acronym, that's fine. Like everyone kind of puts his or her own spin to memorizing a whole wealth of information. There's always flashcards, but it is time consuming, so I do recommend, I personally found it helpful just to write all this out and have my own study sheet, or I'd write on the dry erase board. Okay, I have one more in mode nutrition. I just ran out of room. So we have this is Thermus aquaticus down here, and Pyrolobus, chemolipid autotroph. Okay, just add this to the row below the chemolipid autotroph. Type. It's chemo, organo, heterotroph. So you may not see the organo root word in there sometimes. It may just be chemo heterotroph. It's the same thing. The energy source is organic compounds, sugars. I'm just going to write amino acids, etc. Then the carbon source makes sense, right? Organic compounds. And what makes something classified as organic is those carbon-carbon bonds. And examples of this is E. coli, Bacillus, Pseudomonas, and Streptococcus. Okay, so you should have 4 rows there about the energy and carbon sources used by different prokaryotes, different groups of prokaryotes. Okay, now we're gonna move on to energy carriers. The energy carriers for cellular respiration are NAD and FAD. The reduced forms are NADH and FADH2. For photosynthesis, instead of NAD, you have NADP. The P, you just think photosynthesis that starts with P, NADP, for that carrier. But yes, I'll write this out and then I'll show you just a quick review between oxidation and reduction, just to make sure y'all don't get confused with that. I mean, honestly, there's so many websites, or you could look in your book. I mean, there's just different ways to test yourself, basically. Okay. Energy carriers. So, like I said, you have NADH and FADH2, and NADPH. That's NADPH is for photosynthesis, and then NADH, FADH2 for cellular respiration. These are the reduced. They're in their reduced forms here. So NAD is derived from niacin, and niacin is a form of vitamin B3. And FADH2, or well, I guess just FAD, is derived from riboflavin. So niacin, this is an A. Niacin is a form of vitamin B3, and then riboflavin is basically just also known as vitamin B2. It's usually used as a dietary supplement. Anyway, so energy carriers are electron carriers. Why are electrons so important in this case? Well, on the different pathways like glycolysis, Krebs cycle, okay, or pentose phosphate pathway, these energy carriers, so once NAD comes in, picks up an electron, it's in its reduced form as NADH. It goes to the electron transport chain, to then generate ATP. So we'll be going through how that happens in a little bit. Okay, so energy carriers, NAD, FAD, they are electron carriers, okay? They contain reducing power for cells, NAD and ADP carry H minus or hydride ion, 2 electrons and 1 protein. Reduced molecules generate a proton motive force. So if you have all these reduced energy carriers coming to the electron transport chain, then it's going to generate the proton motive force at the membrane, which is where the electron transport chain is in prokaryotes. So if I write PMF, I'm indicating that for proton motive force. This is important to know there. Okay, now I'm going to show you what page just to briefly review oxidation reduction, and then we'll get back to the transport mechanisms in the cell. Okay, so y'all remember oxidation numbers? Briefly, like if you just read the periodic table, you can get the oxidation number. But oxidation occurs when the oxidation number of an atom becomes larger. So remember oil rig, oxidation is loss of electrons. So whenever that element is losing, or that compound is losing electrons, then the oxidation number increases. Then reduction, or becomes more positive, that's a better way to say it. Then reduction occurs when the oxidation number of an atom becomes smaller. So reduction occurs when the oxidation of an atom becomes more negative, because you can have oxidation numbers that are negative. So basically, oxidation is when the oxidation number of an atom becomes more positive. Reduction is when the oxidation number of an atom becomes more negative. Okay. I thought this was a good example to show you here. So in this case, magnesium loses each magnesium, because there's 2 magnesium here, they lose 2 electrons to form the Mg2 plus ion. So, and then plus those 2 electrons that it lost. So in this case, the magnesium is being oxidized because it's losing electrons, right? Then each oxygen molecule gains 4 electrons to form a pair of, yes, okay, so the 4 electrons here should form that. Because oxygen is normally seen in the form of a diatomic molecule, which is why it has a subscript 2. And, okay. And then since it's gaining electrons, right, it's gaining 4, then it's being reduced to this. Okay? So honestly, like, you need to ask yourself, okay, is this element or is this compound losing electrons or is it gaining electrons? Because basically energy is generated based on the flow or the transfer of electrons, in this case at the electron transport chain. Okay. Now moving on, we're going to do the transport mechanisms into cells. Transport mechanism into cells. So you have, for large molecules, in order for them to get into the cell, there are exoenzymes. Exo, that prefix usually means like to exit or to put out. Secreted by cell proteins. For example, flagella. Yep. Okay, then for the smaller molecules, it's a little bit easier for the smaller molecules to be transported inside. So you have, and I'll mainly be focusing on these, the transport mechanisms for the smaller molecules. You have passive, simple, diffusion. My handwriting usually looks better in person, or maybe that was just me. That's fine. Facilitated diffusion. Facilitated diffusion. Active transport. And with active transport, they're usually these protein transporters or protein families, and I'll talk more about this in a few minutes. Group translocation and siderophores. Okay, that's the broad umbrella topic for the different transport mechanisms. Now I'll go a little bit more detail, especially into facilitated diffusion and active transport. So just to make sure y'all remember this, the simple diffusion, the external concentration is greater than the internal concentration. Basically, if the solute is trying to get in to the cell, then for that to happen, that solute needs to be in the external. It needs to be in the outside part of the cell, like the cytoplasm or the cytosol, basically. And then that external concentration needs to be greater than the internal concentration of the cell, allowing for the solute to simply diffuse through. And that happens until equilibrium is reached. Equilibrium is whenever there's the same amount of concentration on both sides of the membrane. Does not occur for polar molecules. Now, I'm going to talk about facilitated diffusion right now. And as you can see, facilitated diffusion is the same thing where the external concentration for the solute to be able to diffuse through the membrane, or diffuse through the different protein channels, because if you have some of the smaller molecules can't just diffuse through the phospholipid bilayer, they have to go through protein channels. So for those protein channels are facilitating that solute to move through the membrane and into the internal part of the cell. Anyway, so with facilitated diffusion, it's not stopped by inhibitors. Constitutive, yes. So it's always working 24/7. Active transport is the opposite, and I'll get to that. Okay. So I'm just going to denote facilitated diffusion as FD. So FD is a passive process. It does not require energy to occur. That's what I mean by the passive process. It eliminates concentration differences. Facilitated diffusion is more common in eukaryotic cells and prokaryotic cells. The examples where you see facilitated diffusion in prokaryotes is in glycerol transport and in aquaporin. I will draw a little schematic for the glycerol transport, and then I'll talk about aquaporin. Glycerol transport. So in facilitated diffusion, the protein channel allows active uptake kind of open there. So here's the cell membrane. Y'all get the idea. Hydrophilic head, hydrophilic tail. Okay. Cell membrane, cytoplasm. We have glycerol. So this goes through. This protein complex here is called GLPF. Usually proteins start off with a capital letter and they are not italicized. And then this is glycerol in the extracellular fluid. Okay, that happens. So first step, glycerol enters GLPF channel. Next, so now together here. So the same thing as before. Cell membrane, but the positioning of the glycerol is different, etc. Just gonna do etc. there. So cell membrane. So the glycerol is here, and now it's moved out of the channel and to inside the cytoplasm. Extracellular fluid. So here, glycerol is able to move from, glycerol moved into the GLPF channel, right? Then what happened with these proteins, a conformational change occurred, which then opened the channel to the other side, allowing glycerol to move into the cytoplasm. So a conformational change in GLPF opens the channel in the cytoplasm, and glycerol enters the cell. Okay, that pretty much covers the glycerol transport. Now I'm going to move on to aquaporins. Now I've been talking about glycerol transport and aquaporins because these are two examples seen in prokaryotes for facilitated diffusion. Okay. If you want to put facilitated diffusion in parentheses, just remind yourself, we're still talking about facilitated diffusion, that's fine. Okay, so aquaporins, they were first described by Peter Ager, and he won the Nobel Prize for his research on aquaporins. You can probably add this to the history handout that I showed y'all in the first two sessions. Okay, so aquaporins belong to the major intrinsic protein family, so MIP. Aquaporins allow water or urea to come into the cell. Aquaporins are found in mammals, plants, and quite a few bacteria, but not all of them. But not all bacteria. Bacteria have a single type of an aquaporin protein and the examples for facilitated diffusion under aquaporins are with the protein complexes is AQPZ, AQPX, and GLPF. So the GLPF is in glycerol transport, and AQPZ and AQPX for UC and aquaporins. Examples are AQPZ, AQPX, and GLPF. Okay, so it's aquaporins. And pretty much with facilitated diffusion, so glycerol transport aquaporins, they fall under facilitated diffusion. Pretty much in facilitated diffusion, the solute goes with the gradient. So you can say facilitated diffusion, solute, or you can say particles go with the gradient. Or you can think go with the flow if that makes it easier than to say gradient. Okay, now active transport. You can always think that for active transport, the particles need to be like, they're still going to be moving, but they need something to kind of actively transport them. They can't, facilitated diffusion doesn't require energy to transport the molecules, but an active transport energy is required. So you can always think like active transport, the A, and think ATP is necessary. So energy used to move molecules against gradient. That's why the energy is necessary. The energy is necessary because those molecules are trying to move against the gradient. So you need energy to combat that gradient. ATP or proton motive force. Okay. So yeah, you pretty much have hydrogen ions ejected from the cell. Hydride ions remain inside the cell. The separation of charge creates an electrochemical gradient. And this gradient is controlled by the controlled flow of hydrogen ions back into cell, release of energy. Okay. So what's happening is that you have these proteins with the membrane. So lipid bilayer membrane, right here. Okay. This, so whenever, whenever the energy carriers are coming to the membrane, dropping off those electrons, right? Those electrons are going through these protein complexes here along the membrane. The hydrogen ions get kicked out of the cell. This is outside, this is inside. Oh, inside. And the hydride ions are in the cytoplasm. And then eventually we're going to hit ATP synthase. I'll get to that in the next video. But just so you know, the proton motive force the proton pumping action of the electron transport chain results in a proton motive force. And proton motive force is an energized state across the membrane resulting from a proton gradient. During respiration, this proton motive force is used by the ATP synthase to make ATP. So the ATP synthase can't phosphorylate an ATP to make an ATP unless proton motive force is present. Okay. Now, what goes hand in hand with active transport are the ABC and MFS transporter proteins. So ABC, you'll learn another transporter protein around whenever you talk about antibiotics. The ABC transporter stands for ATP binding cassette protein. MFS is major facilitator superfamily. And so the ABC transporters consist of a 6x6 structure of helices. I'm just saying this because of its structure. I don't really know, at least in the course that I assigned for with the specific professor, we didn't really go into too much detail on the structure. The main thing that you need to know about ABC and MFS were what those transporters use to pump the molecules against the gradient. So ABC uses ATP. So focus more on what these transporters uses to pump the molecules against the gradient. If I say versus, I mean against the gradient. Okay. MFS also has a 6x6 helical layout. Just so y'all know, the helical layout, or the 6x6 thing, what that means is, you might, okay. 6x6, so 1, 2, 3, 4, 5, 6, and then there might be a protein complex here or some sort of loop domain, and then I'll go back 1, 2, 3, 4, 5, 6. So that's what it means by a 6+6 helical layout. So both ABC and MFS have that, but just whenever that loop goes down, whether there's a protein complex there or a different, or like a binding motif or something, that's what can differentiate between the two. But like I said, worry more about the function of the transporters, not so much the structure. I just added the structure to be more specific. So mated facilitator superfamily, MFS, uses proton motive force to pump against gradient. So there are 3 types. You have importer, which is where 2 molecules come into the cell, antiporter, where 1 molecule comes in, 1 goes out. So symporter, you can think like symmetry, same. Anti means against, so like they literally are going, or in that case, one's going against each other while one's going in, one's going out. And then uniporter, just think uni, like unicellular, meaning 1. Okay. Now after talking about this with facilitated diffusion and active transport, I have 2 videos I want to show y'all. I can't post it with the handout because you need an account to log in. So I will show that to you now, and I think it'll clarify some things.
The formal definition of a concentration gradient is the process of particles, which are sometimes called solutes, moving through a solution or gas from an area with a higher number of particles to an area with a lower number of particles. The areas are typically separated by a membrane. This membrane can be permeable, semi-permeable, or non-permeable. Permeable is defined as a membrane that can be crossed by particles, ions, or water. Semi-permeable means that some particles, ions, or water can cross the membrane. Finally, non-permeable membrane means that no particles, ions, or water can cross the membrane. An example that might help you understand the different types of membranes would be different types of fences. A wooden log fence would allow many things to pass through. This would be an example of a permeable membrane. A chain link fence would allow some small items to pass through it. This would be like a semi-permeable membrane. A solid plastic fence would not allow items to pass through it at all. This would represent a non-permeable membrane. Solutes moving through solution, that is, a liquid or a gas, happen by random motion until there are equal numbers of particles in the 2 areas. Random motion is defined as movement that occurs by chance because there is no order or regular system by which the movements of the particles shift in the solution or gas. The actual movement is called diffusion. There are 3 types of diffusion: passive diffusion, facilitative diffusion, and active transport. Because active transport does not happen along the concentration gradient, it will not be covered in this lesson. Instead, let's first discuss passive diffusion. Passive diffusion does not require energy. It happens by random motion. In a solution or gas that has an area of high numbers of particles and an area of low numbers of particles, the particles will diffuse or move from the area of higher to the area of lower concentration. A common example of this is a cup of water that you drop food coloring into. The food coloring is concentrated when dropped into the cup of water. However, after a few seconds pass, the particles become lighter in color as they move to the lower concentration level. Again, this happens without energy, so it is known as passive diffusion. Another type of diffusion that is passive because it does not require energy is facilitative diffusion. This is how particles move across a membrane. This usually refers to a cell membrane, so that is the description we will use. A cell membrane is a barrier that usually cannot be crossed by all particles or ions. Ions are just charged particles, which means they have either a positive charge or a negative charge. The particles cannot move freely across the membrane into and out of the cell. They must be assisted by a carrier. So this is called facilitation. This carrier is usually a protein that is able to cross the cell membrane. It has a special shape and usually can carry a small particle or ion across the cell membrane through a specific channel. This process can either happen at any time, or the channel may be opened and closed at different times, depending on the cell's needs. To assist you with this explanation, think of the process of a person having surgery. The person having surgery cannot go into the operating room. They must first be taken into the surgery suite. Also, the surgery door would not always be open to the person. The doors would only be open when they are scheduled for surgery. In addition, patients cannot go into surgery by themselves. The nurse takes the person through the operating room doors. Do you see how this relates to the process of facilitated diffusion? Remember, the protein carries the particle or ion across the membrane through a channel in the cell membrane wall. The concentration gradient is an important process for understanding how particles and ions move in random motion in a solution or gas. It is the process used for particles moving from an area of higher concentration in a solution to an area of lower concentration. This occurs until there are equal parts of particles in the solution or gas. Processes that use the concentration gradient include passive diffusion and facilitated diffusion, which occur without the assistance of energy. Passive diffusion happens by random motion, while facilitated diffusion requires the assistance of proteins that travel through channels in a membrane.
Okay, so that was the first video, and then the second video is
Have you ever been to the fair when it's really crowded? Well, maybe you wanted to go, but it's too crowded for you. So the lines are too long, and the people are shoulder to shoulder, carrying cotton candy and spilling popcorn all over the place. Perhaps you wait in the busy parking lot, observing the throngs of people and wondering if you shouldn't wait till it's not so busy. You're not a fan of the crowds, but you are a huge fan of the fair. So finally you decide, let's face it, it's a fair. A fair is always busy. There's only one thing to do in a situation like this. You muster up all your energy, and you fight your way through the admission gate to get yourself a roll of tickets and some fried dough. Does this sound like the town fair? Well, there sure are a lot of things going on in this cell at the same time, so it might seem busy like a carnival. There's also many times when a cell needs to transport something in that goes against the concentration gradient. So let's think of the fair like a concentration gradient. Remember that a concentration gradient is the gradual difference in solute concentration between two areas, much like you standing outside the fair while there are thousands of people inside. There is a higher concentration of people inside the fair. So when a cell needs to transport something along a concentration gradient or from an area of high concentration to an area of low concentration, it does this by passive transport. This would be like people leaving the fair because it's crowded. That's easy. However, when a cell needs to transport something against a concentration gradient, or you decide to move from an area of low concentration of people to an area of high concentration of people, this is going to take some willpower, or in the case of the cell, some chemical energy. Active transport is transport against a concentration gradient that requires chemical energy. Active transport moves ions or molecules in a specific direction through the use of an integral membrane protein. Think of these like different gates at a fair. There are three types of membrane proteins that use energy to push these substances against the concentration gradient. A uniport is an integral membrane protein that moves an ion or molecule in one direction. Uni can remind you of the word one, such as one substance moving in one direction. This would be like an admission gate that people could only enter, or an exit gate through which people could only leave. An antiport is an integral membrane protein that moves one ion or molecule in one direction while moving a second substance in an opposite direction. The anti in antiport means against, and can remind you of two substances moving against each other, like an admission gate that only lets a few people enter if a few people exit at the same time. A symport is an integral membrane protein that moves two ions or molecules in the same direction. Sym in symport stands for the word same, as in two substances moving in the same direction. This would be like an admission gate that had two lines of people to enter the fair at the same time, in the same direction. Both antiports and symports are a type of coupled transport, because they transport two different substances through the same integral membrane protein. Importantly, all three different types of integral membrane proteins use chemical energy to make this active transport happen. This allows a cell to maintain specific concentrations of substances different than in its surroundings. Let's talk about a specific example of active transport that is going on in all of your nerve cells right now. Your nerve cells require that a specific concentration gradient of sodium and potassium be maintained in order to properly conduct your signals. There is a higher concentration of sodium ions outside a cell than inside a cell. There is also a higher concentration of potassium ions inside the cell than outside the cell. The sodium-potassium pump ensures this gradient exists by constantly pumping sodium ions and potassium ions against the concentration gradient using active transport. First, the sodium-potassium pump in the cell membrane binds 3 sodium ions from inside the cell. Next, with the help of chemical energy, the sodium-potassium pump changes shape and exports these sodium ions outside the cell. This is against the concentration gradient of sodium. 2 potassium ions from outside of the cell then bind to the pump and are imported into the cell. Again, this is against the concentration gradient. Because 2 substances are transported in opposite directions, like the Baire gate that is both an entrance and an exit, the sodium-potassium pump is a type of antiport. In summary, we've learned that active transport is transport that requires chemical energy to move substances against the concentration gradient. There are 3 types of integral membrane proteins that aid in active transport. Uniports are proteins that move one substance in one direction. Antiports are proteins that move 2 substances in opposite directions. And symports are proteins that move 2 substances in the same direction.
Okay, now I'm going to go back. So that was to cover active transport and facilitate diffusion. Now I'm going to move on to siderophores. So my question was, why do less than 1% of all bacteria in the environment grow in lab media? Specifically, it is 0.4% of bacteria, of all the bacteria in the environment. Okay, of all the bacteria in the environment, only 0.4% of that bacteria can be cultured in lab media. Why is that? Well, there's a hypothesis. I can't pronounce the names of the scientists, but there was a hypothesis in 2010, and their hypothesis was that bacteria lack the ability to produce siderophores, and for siderophores in growth, the bacteria requires bacterial interactions with neighboring microbes growing within communities. Those siderophores are basically all these characteristics about siderophores here. They are produced by microbes. They bind iron, and they pass that iron to they pass through transport proteins and then hand off that iron that they were carrying, or they pass the iron to the transport proteins. Some of the siderophores are ABC transporters. Other bacteria bind human iron complexes and take iron from them. Okay, so I'm going to draw a picture with talking about the siderophores and iron transport. But I just wanted to make sure you all have a chance to write this down. But basically, whenever you think of siderophores, just think that it's produced by microbes, it binds iron, and it can either pass that iron to the transport proteins, or it goes through the transport proteins and then releases that iron, and that iron then binds to other complexes that are inside the cell. Pretty much it helps facilitate that iron to get into the cell. Okay. Anyway, so siderophores and iron transport. Okay. So first, E. coli synthesizes and secretes iron binding So this is the this is acting as the siderophore in this case. Okay. So we have the Fe3 plus and then the enterokinin here. And they pretty much come together to form this. Yep. So this then goes into the cell, or cell membranes specifically. Outer membrane. So this goes through this protein complex called that A. A goes through, and then we have E. Okay. Then here we have the second step. Complex is transported to the periplasm. They're talking about periplasm, you know, this is going to be in a gram-negative cell, through an outer membrane, outer membrane protein. And that outer membrane protein here is A. Okay, then I guess you can say this is 2A and then 2B here, protein. So this complex is then transported into the cell by an ABC transporter that includes a periplasmic solute binding protein, which is that B. And integral membrane protein, which I will draw in just a minute, that C, that G, and D. Okay. So these two come together, right? So it's now looking like this. And now this enters the with these protein complexes. So this is that G. That C, that C, I'm drawing this just to show you that this is the outer membrane, or sorry, cell membrane. We did outer membrane before. So this would be like the inner membrane, if you want to call it that, but I was just going to say cell membrane. So FEP-C, then now The step B helps the complex to enter into the cell, and by the time it goes through the step B and step C, you just have the original complex left over, but you have ATP being dephosphorylated into ADP plus phosphate. Okay. And then now that you have this, right, I'm just going to arrow over here. What you have right now is this. And then once it's inside the cell, then the siderophore releases the iron. So now you're back to the individual. Okay. Oh, I'm sorry, the iron is released to Fe2+. So inside the cell, the iron is released, and the iron is reduced from Fe3+ to Fe2+. Okay, I think that is pretty much all I wanted to say for that page. Now, just a few more topics left, and then we'll be done with this session. Group translocation. Group translocation. This is important for glycolysis. Group translocation occurs at the beginning of glycolysis whenever, let's say, glucose is trying to get into the cell. Okay. An example of group translocation is with the phosphotransferase system. Basically, group translocation is important in glycolysis. So if you want to just say, like, define, group translocation is a process in which an organic molecule, like glucose, like I just said, as glucose is transported into the cell while being chemically modified. Specifically, the organic molecule binds with the protein on the cell surface, and then once that molecule is bound to a protein on the cell surface, then that's whenever the chemical modification occurs. So followed by altering the molecule's chemical structure, during its passage across the membrane. So example is phosphoenol pyruvate PEP, and that is also on your handout, your metabolism handout. Oil rig, I talked about that earlier. PEP, phosphoenol pyruvate to pyruvate. Okay. PEP phosphotransferase system. So that's kind of like the definition, and I'm just going to add a couple more bullet points for the section. Chemical alteration of molecule during transport. So I kind of said that. It was just in the parentheses. I just wanted to repeat it just in case. Transport across the membrane. And normally that chemical modification is adding a phosphate. That's usually pretty much what's happening. Usually add phosphate. I just do P with the circle on it. That means phosphate. This does not affect the concentration gradient. Concentration gradient. And this usually occurs with sugars such as glucose, fructose, and arabinose. Also some other sugars, but usually those three. Yep. That's pretty much all I have for group translocation. And okay, so this whole time we've been talking about transport in prokaryotes, and I'm going to briefly just talk about the transport in eukaryotic cells. It's pretty similar to prokaryotes except for two things. So transport in eukaryotes. Usually the sign is similar to in math. So transport in prokaryotes, you have the simple diffusion, you have facilitated diffusion, you have MFS, ABC. So you have active transport. Now the difference here with eukaryotes and prokaryotes is endocytosis and exocytosis. Eukaryotes only. So with endocytosis and exocytosis, you have pinocytosis and phagocytosis. Those are two. They're very different. Endocytosis, pinocytosis is the process by which eukaryotes take up material. Okay, so how they do that is the extracellular molecules, so the material that the cell ultimately takes up, first binds to receptors. Clathrin internalizes and then forming the endocytic vesicles. There's a picture of this in Nestor. And then endosome with endocytic vesicles. It's an endosome that makes an endosome, and the endosome fuses with the lysosome. And remember, lysosomes carry usually enzymes within the lysosome, and once an endosome fuses with lysosome, then those enzymes help degrade whatever material within the endosome. Okay, then phagocytosis. Pseudopods surround material from the outside, from like external to the cell, and then internalize that material as the phagosome, and then that phagosome fuses with the lysosome to form the phagolysosome. Yep. Like I said in the previous videos, just focus more on the prokaryotes. I just briefly touched on the eukaryotes just to show you the brief differences. So that pretty much wraps up this session, and I will be doing metabolism part 2 for the next video. Okay. So thank you. Good luck studying.